And thanks, Emilia, for getting me here. And thanks all for coming. I'm really excited about the opportunity to share my work here. And today I'm going to share a latest project of mine on estimating the impact of Neanderthal introgression on human complex traits. And as Kelsey said, I'd welcome questions in between, and if there's any clarification needed or anything. And this work hasn't been submitted yet, and I would really love to get some comments and feedback from you. And yeah. So the question we're going to ask is really about the impact of Neanderthal introgression in present-day humans. About 50,000 years ago, modern humans migrated out of Africa and encountered and mixed with the archaic hominid Neanderthals. Genetic analysis have revealed that present-day non-African human populations inherit 1 to 4% of their genetic ancestry from the Neanderthals. Because of the high divergence between the humans and the Neanderthals, this introgression event introduced many novel variants into the ancestral out of African human gene pool, which may have helped the human population survive the new environments they encountered because the Neanderthals were there before humans. In principle, studying the Neanderthal-derived mutations in large cohort of individuals measured for diverse phenotypes can help us understand the biological impact of Neanderthal introgression. And the largest accessible cohort that can allow such a study is the UK Biobank. It is a British cohort which contains about 500,000 individuals, and these individuals are genotyped and they are also measured for thousands of phenotypes. This database allows researchers to investigate a wide range of questions, and in our case, we're interested in introgression. One way of understanding the impact of introgress variants is to perform an association study on the introgress variants, which is basically a linear regression analysis between the SNPs and the phenotype. And previously, Denman and Kelso showed that some Neanderthal introgress variants are significantly associated with traits, such as the skin tone, by using this approach. However, in a different study, using the data from an Iceland cohort, Skalb and the colleagues suggested that most of the associations at the Neanderthal introgress SNPs were driven by the associations at linked non-archaic variants, indicating that there might be a limited contribution to modern human phenotypes from the Neanderthal introgression. However, association analysis for single locus ignore the linkage disequilibrium structure, the LD structure, and cannot settle this debate. This is because in simple GWAS analysis, the correlation among loci are not taken into account. Each time only one SNP is being regressed with one phenotype. And this correlation is just this linkage disequilibrium. So in this example is like a linkage between two allele scenarios. So there are two locus in the genome, and each locus have two allele, locus A and locus B. And there are 4 possible haplotypes, and when they are in equilibrium, the locus A and locus B are basically independent from each other. Therefore, the frequency of the haplotype with both A and B could be predicted by the allele frequency of A and allele frequency of B as a simple product. But when there is linkage disequilibrium, for example, if A1 allele tend to co-occur with the B1 allele, this product rule no longer holds. And this means that locus A and locus B are correlated. And the correlation between the loci makes it harder to differentiate which mutation is truly associated with the phenotype when a single locus association is used. And in statistical genetics, people just often use this square of correlation coefficient, or R square, to measure this linkage disequilibrium, This is a concept that we will come back with later. So that side, single locus association is one way to approach this problem. And another possible measure is to quantify the aggregate contribution of introgress Neanderthal SNPs to trait variation. And in this case, people would like to measure something about heritability. Narrow sense heritability is the total phenotypic variance explained by the genetics in a population through an additive model. And in terms of the impact of introgress variance, people are interested in something like introgress SNP heritability, which is basically the fraction of phenotypic variation explained by introgress variance based on an additive model. And previously, recently, really in this year, there's a study by MacArthur and colleagues. They employed something called stratified linkage disequilibrium score regression, or stratified LD score regression, to estimate the proportion of heritable variation that can be attributed to the introgress variance. And they found that Neanderthal ancestry is significantly depleted for the heritability of most of their studied UK biobank phenotypes, but is also significantly enriched in some phenotypes. And this equation I'm showing here is the equation for LD score. For each SNP, it computes the sum of the R square with all the other SNPs within usually a 10 megabase pair region. And this quantity, it has some mathematical relationship with the GWAS statistic when the GWAS statistic is computed for each locus. And when people regress this quantity with the GWAS statistics, such as chi-square, it will give people the heritability estimate. And this stratified LD score approach is basically a more advanced version, which basically compute these stratified LD score for a particular group of SNPs, such as the introgress variants. And then it could provide the per SNP heritability for each introgress, for the average introgress variant SNP heritability, and extrapolate the total introgress heritability. And this approach has an advantage, that is, it can start with GWAS statistic, which can either be downloaded somewhere or it's very fast to acquire. And this LD score is usually computed by an external cohort, which is smaller than the study GWAS data cohort. And it also comes with some downside. For example, one particular thing relevant to this study is that it will only work with SNPs that have high minor allele frequency, such as 5% frequency, which is the suggested threshold. And additionally, this mathematical relationship, it holds when the data for LD score computation and the data for regression are matched data. And it's not necessarily true when this LD score is computed from an external data set. So while both studies are really interesting, none of the previously mentioned studies evaluated the statistical power and calibration of their methods. The unique demographic and evolutionary history of the archaic introgress variants actually results in distinct genomic distribution and population genetic properties that makes statistical genetic analysis more challenging than usual. And therefore, it's necessary to account for these factors and sufficiently evaluate the statistical power and calibration of any statistical genetic methods applied to these introgress variants. And to start with, the admixture fraction is believed to be very low, and it's still a debate whether it's 2% or it may start as high as 10%, but gradually decrease to 2%. But in any case, the admixture fraction started low, And there are potentially only very few Neanderthal haplotypes admixed into the human gene pool. And most of the uniquely Neanderthal mutations started at a low frequency, which is supposed to close to the admixture fraction. Without any directional forces such as selection or dilution, they are expected to stay at the same frequency as they enter. Because of this, most of the Neanderthal informative mutations, which we call as NIMs, names, still segregate at low frequency in present-day human population. And in this graph, I'm showing the cumulative minor allele frequency distribution of Neanderthal informative mutations names compared to the modern human SNPs. And the names are shown as this green curve, and the modern human SNPs, this pink curve. We can see that these two distributions are very different from each other because they will undergo different demographic processes. And there are more modern human SNPs. There are more modern human SNPs with ultra low frequency, but they also have ultra high frequency compared to the Neanderthal SNPs. If we use this 5% math as the cutoff, we get about 27.7% of names above this frequency, in contrast to more than 40% of SNPs. In any case, if the study is done with high frequency names, we're going to miss the majority of Neanderthal introgress mutations. And further assumptions have to be made to assume that names with high frequency tend to have the same property as names with low frequency, and it is something that's not known. Similarly, I computed their LD score for the names and modern human SNPs, and that basically estimates the overall linkage disequilibrium effect with the other mutations in the genome. In this graph, I'm showing the cumulative LD score for names and for modern human SNPs. And we can see that names tend to have much larger LD score compared to modern human SNPs. And this again is a signature of introgression because these Neanderthal mutations, they introgress into human gene pool within a few haplotypes, and they tend to stay in strong LD with each other. Only recombination will break it down. And just given the generations since introgression, this is the remaining linkage disequilibrium that's still very strong. I think I see a question from Amelia. Please feel free to unmute or.
Hi, April. Sorry, you said we could ask you questions in the middle, right?
Yes, yes.
Okay. So the LD score measures, so it takes, I guess I'm trying to understand what's on the x-axis. So that LJ, that's a summary of, is it all the pairwise?
Yeah.
So yeah, this LD,
Sure,
this LD score, so let's assume this mutation is J. So it's basically the R square, the square of the correlation coefficient with all the other mutations that's in the neighborhood. And the neighborhood is only the 10 megabase region.
Okay, so it's done per window.
Yeah, it's some sufficiently large window, I would say, because 10 megabase is like 10 centimorgan in general, which basically, yeah.
Okay, sorry, I thought it maybe was like one measure for the whole gene, but it's just in these 10 megabases. Okay, thank you.
Okay. So if there's no more question, we have the LD score for the names and modern human SNPs computed, and also their math. I'm just going to show you that this introgression also influences the coupling between the minor allele frequency and linkage disequilibrium. So here on the left-hand side, I plot the allele frequency against the LD score percentile, and here is using 20 to 100 percentile against the math within that 20th percentile. And on the right-hand side is the MAF percentile against the LD score within each group. And names are in green as before and modern human in pink. The names on the left side. So distinct from the modern human SNPs, this MAF and LD score of the introgress mutations tend not to increase with each other a lot. But in modern human SNPs, there's a strong pattern that's because it's from de novo mutations. So this demographic process affects the coupling between MAF and LD. In statistical genetics, there have been some work showing that per SNP heritability on traits can be allele frequency and LD dependent. Therefore, these are the factors that we need to take into account. Both the marginal effect of MAF and LD, but also the coupling between them. And the goal of this study is threefold. As mentioned, it's important to test the statistical method because calibration may change. So first, we want to identify a suitable method for understanding these introgress variants through realistic UK biobank size simulations. And secondly, we'd like to test some hypotheses. In particular, the null hypothesis would be that Neanderthal variants have the same contribution to phenotypic variation as the modern human variants. And that is after controlling for the other factors such as MAF and LD. And we want to do this across a wide range of interesting phenotypes. And lastly, if possible, we'd like to pinpoint regions of the genome or mutations that introgress variants modulate these complex traits. And to do this, we first need to get a list of Neanderthal introgress mutations in the UK biobank data. And this is the time to give a shout out to my postdoc advisor, Sriram Shankaraman, who added 6,000 SNPs to tag the Neanderthal informative mutations in the UK biobank exome array in 2015. Without that effort, this project wouldn't be possible. He started with about 95,000 Neanderthal introgress mutations from his previous paper that is basically a previously compiled map of Neanderthal haplotypes in the 1,000 genome European populations. And the criteria was basically something like these names have to perfectly match the LT allele in the Europeans, and they're totally absent in the 1,000 genome African populations, which is quite stringent. And after removing the names that are well tagged by the SNPs previously in the UK biobank belief array, that was the first array they used to genotype some portion of the UK biobank data. And he used a greedy algorithm to select about 6,000 names that further tag all the remaining set of SNPs at R² 0.8. Therefore, all these names, they're being tagged with the genotyping array. And then these 6,000 names are designed into array, and they're being used in all the UK biobank data. That's the 5,000 individuals. And this data was released the genotyping data was released in 2017. And because these arrays are designed to tag all the names from the whole genome, they can allow good imputation for all the names genome-wide. And the imputed data was released 2018. So we first perform a quality control on the imputed genomes and the SNPs and annotate the QC SNPs by ancestry into Neanderthal informative mutations, names, and modern human mutations. And we also annotate them by allele frequency and LD score. As was previously mentioned, names tend to have higher LD score and lower allele frequency. Statistical genetic analysis designed for general mutations may not calibrate the same way when applied to names. So in order to evaluate this method, we need to have large-scale realistic simulation. And in this case, I simulated phenotypes based on the same set of UK biobank genotypes we use with different levels of polygenicity, heritability, and the coupling between effect size, MAF, and LD. High heritability simulations have heritability equals to 0.5, and moderate heritability simulations have heritability equals to 0.2. And since we're interested in complex traits, we focus on moderate and high polygenicity with either 100,000 SNPs being selected as causal variants, that's about 1%, or 10,000 of the variants as moderate polygenicity. And I simulated 5 different genetic architecture, which we name as baseline, common, rare, high, and low. Baseline considers the SNP effect size are neither MAF or LD dependent. So they're just randomly sampled SNPs in the genome. And common and rare are allele frequency dependent architecture with either rare variants being enriched for phenotypic effect or common variants being enriched in phenotypic effect. And high and low are LD dependent architecture with either high LD score SNPs enriched for phenotypic effect or low LD score SNPs. And in all these simulations, names are neither enriched or depleted in heritability. So ideally, a method shouldn't differentiate, shouldn't detect enriched or depletion when applied to this simulated data. And because we have the causal names, we can also compute the causal name heritability by basically doing the sum of the beta square of the names divided by the total phenotypic variance. And this could be used to benchmark with the estimated name heritability. And for today's seminar, I'm only going to show the methods that are tested to be calibrated. So the method I eventually use is RHMC, which is a method developed by my lab mate Ali, which was just published a year ago. This is a fast and scalable multi-component HE regression method, and with the idea behind that SNPs can be divided into different components, such as their K components here. And the SNP effect size for each component is different. So this sigma square is different for SNPs belonging to each component K. And this sigma square will be estimated with our RHMC given a K group that is user defined. And with this, we can have total SNP heritability for each group, and we can also combine them into total heritability or combine them into different components we're interested in. We want to test whether name heritability is higher or lower than the modern human heritability. And the simplest naive approach is to design something with 2 components, one for Neanderthal mutations names and another for modern human mutations, which is defined as the rest of the mutations that are not annotated as names. But because, as mentioned, different allele frequency and LD score could also have an impact, we also have annotations based on ancestry plus allele frequency and ancestry plus LD score, or ancestry plus allele frequency plus LD score. And we use non-overlapping annotations, which means that one SNP will only belong to one annotation. And this allows us to combine the heritability estimated from each component into a mega component. For example, there could be 5 components for the Neanderthal introgress mutations. Here, when we use ancestry plus MAF, we can combine them into one component after estimating the heritability for each component, which is basically what's here the total name heritability is the sum of the heritability of names that's in each component for names. And because we want to compare this quantity with the modern human SNPs heritability, and the number of names and the number of modern human SNPs falling into each component could be each MAF and LD component could be very different. And here I'm defining the modern human heritability as a related heritability, which is basically by normalizing the number of modern human SNPs and correcting it with the number of Neanderthal SNPs, such as these two quantity will have MAF or LD or MAF and LD being controlled. And when in our simulation, these quantities are of interest, but we also want to test if they are significantly different from each other. And therefore, we define the delta heritability as the name heritability minus modern human heritability. And Ali made an extension of RHMC for this project, which will also be released together with when we preprint it, which is basically to also allow the standard error estimate being estimated for this user-defined quantity. So with the standard error for each quantity also estimated, we could convert this into a z-score by dividing this quantity with the standard error and then perform hypothesis testing. So at first, benchmark the calibration of RHMC with the simulations mentioned before. We have 5 different architectures as listed here, and I also combine them into this, combine them together into all. And in each panel here, there are basically 12 simulations with different levels of polygenicity, heritability, and replicates. Additionally, there's different ways of partitioning, of annotating the genome by ancestry and other stuff. And in this test, we first basically test if the estimated name heritability is statistically indifferentiable from the simulated name heritability. And the shaded area is z-score between +2 and -2. And we can see that for name heritability itself, it doesn't matter that much. Most of the ways of partitioning the SNPs would work, and they're all calibrated. More importantly, we'd like to test whether the estimated name heritability is different from the matched modern human SNP heritability using this delta H2. For this test, again, we expect a calibrated method to have z-score within ±2 as this narrower shaded area. And we find that when we partition by ancestry and MAF and LD, the calibration is very well for all the simulated architecture, and this box is basically within this shaded area. And for common and rare, they are allele frequency dependent architecture, and we see partitioning by ancestry plus MAF would give us calibrated result, but not controlling for MAF doesn't give us calibrated results, such as ancestry and ancestry plus LD. And high and low are LD dependent architecture, in which we see that when we control for LD, such as ancestry plus LD and plus MAF and LD, we get calibrated result, but not controlling for it would yield some biases. And this is basically why we choose these more complicated annotation when we analyze the real data. And lastly, I just also want to show you that in simulations with this partitioning, the total heritability H2 is still calibrated, as we can see here, that they are calibrated for this comprehensive annotation. And in the next few slides, we will see some real data when we apply this approach to the UK Biobank phenotypes. In this study, I used a total of 96 phenotypes from the UK Biobank, and these phenotypes belongs to 14 categories that are listed here. And before we dive into the hypothesis testing part, I just want to show you that these not raw numbers of the heritability and name heritability and delta heritability. On the x-axis is the phenotypes, and each phenotype is 1 dot. Y-axis is on the upper panel is name heritability, lower panel delta heritability. And if you can see this line is, this horizontal line is 0 here and 0 here. So overall, for all these phenotypes, the name heritability is generally larger than 0. In some phenotypes, the error bar is large, but most of the things they're above 0. And for this lower panel, most of the things are below 0, although there are also some phenotypes which are spread out around 0. And this is just an overall look. And we can also use z-score to quantify this and perform hypothesis testing. And in this figure, I'm showing the phenotypic category on the x-axis, and each phenotype is still 1 dot. Some phenotypic categories have more phenotypes, some have fewer. And this shaded area is the ±2 z-score area. So across all the tested phenotypes, when we just look at the z-score individually, there are only 3 phenotypes with significant positive name heritability after multiple testing. And they are overall health rating, waist-hip ratio, and gamma glutamate transferase. And this is potentially because the fraction of names is very small, and the test is slightly underpowered. Within each category, we perform a meta-analysis under the assumption that this heritability within a category comes from the same random distribution. And I'm showing the meta-analysis z-score for the categories here as this bar. And this meta-analysis improved the power, as we could see that most of these tests, they are significantly non-zero for all but 2 tested phenotypic categories, and meaning that the name heritability is generally non-zero. And so next, we'd like to test whether the name heritability is smaller or larger than the modern human heritability. And in this case, per phenotype wise, we find that there are 14 out of 96 phenotypes have a significant name heritability depletion, which is, you can see some of the dots here, they're lower than -3. And most of these phenotypes, as significant depletion phenotypes as listed here, they belong to the anthropometry category. And we again perform the random effect meta-analysis, and we found that we found that there are significant name heritability depletion in 4 phenotypic categories. And in the other category, even after meta-analysis, we don't detect this significant depletion. And this is somewhat surprising. In addition to that, with all the tested 96 phenotypes, we didn't detect any significant enrichment either per phenotype wise and also within category. Although this is consistent with the understanding that there's a lot of purifying selection on Neanderthal ancestry, since the admixture such that they may not contribute as much to phenotypes. Therefore, we identify depletion but not enrichment. It's also, this result is also different from the previous MacArthur paper using SLD score regression, because they've identified both and a lot of depletion, more depletion than what we found here. And we also for the majority of the phenotypes, they're just neither depleted nor enriched, and at least statistically, and suggesting that there could be still some amount of phenotypic variation contributed by the introgress variants. And just to show that the previous result was the result when we control for ancestry and MAP and LD. And just to show that it's necessary to control for this, this plot is the result in real data analysis, with one way is to control for MAP and LD, another way is ancestry only partitioning that is not controlling for MAP and LD. We would get very different result. In this case, each dot is one phenotype, and we can see almost all the dots are below this diagonal line, and even the error bar don't extend there very much. We could see that if we control for MAP and LD, here's 0, and the dots are more or less spread around 0 and not going much lower below 0. But if we don't control for MAP and LD, here's 0, and we would get a lot more significant depletion for name heritability. And this, if we don't control for it, this signal should be fictitious. Additionally, not controlling for MAP and LD also lead to name heritability, lower name heritability, as we could see that most of these dots are below the diagonal line. This and the previous result, previous slide shown, both emphasize that it's necessary to control for these factors when doing hypothesis testing in the real UK Biobank data. In addition, it also suggests that names possibly have different per SNP heritability when they are at different MAP and LD, such that if a study only used MAP larger than 5% to estimate name heritability, it may not extrapolate to the rest of the names in the genome, which have lower frequencies. And in our result, we found that names are not necessarily significantly depleted in terms of heritability. And this is also interesting because it opened up some opportunity to identify mutations with causal phenotypic effect. As was previously mentioned, single locus association tests cannot really control for linkage. A SNP could become significantly associated due to its linkage with a modern human mutation or due to its linkage with a Neanderthal mutation. In order to pinpoint causal mutations, we need to control for linkage. In addition, most significant locus is not necessarily causal. As shown in this figure here, this is a figure taken from the Suzy paper from Wang et al. So in this figure, SNP 1 and 2 in red, they are causal variants. And this yellow colored SNP is the most significant SNP. This is because the complicated LD structure could create this pattern. And complicated LD structure and strong LD among names is a feature of introgress variants. Therefore, we take advantage of this newly available Bayesian statistical fine mapping approach, which is actually designed to tease out these complex LD patterns to apply to the names. This Suzy software, it output something called credible set, which is a set of SNPs has probability greater than a predefined parameter row to contain at least one SNP with non-zero regression coefficient. you can understand it as it will provide a list of SNPs where one of them, at least one of them, is likely causal. A great thing about SUSY is that it uses variational inference to speed up this method, and it's actually applicable to large sample size, such as the UK Biobank. As we're particularly interested in identifying causal names from the entire genome across a large number of SNPs, but SNPs from a credible set of SUSY could be either a name or a modern human SNP or a mixture of both. We took a slightly different approach than just applying SUSY. So here's a demonstration of the fine mapping protocol I use. On the left-hand side is a genomic region with the modern human SNPs in pink and Neanderthal mutations in green. And we first perform a GWAS analysis to get the significant names. As you can see that there are fewer green dots here than here. And to further reduce the number of tests, I do LD pruning and have pruned in significant names. And for example, if we have 3 pruned in names in this region, as illustrated here. After that, for each significant pruned in names, I take the 200 KB region surrounding it and input this data to SUSY. Because we're particularly interested in controlling for the LD with both names and the modern human SNPs, this input genetic data to SUSY contains all the SNPs in the region rather than just the names. And in this case, we're performing 3 different fine mapping analyses, and the output would be credible set if there are credible set. And let's say we get 3 different SUSY output, SUSY credible set, and to get more evidence that the causal variant is names, we only take the credible set with majority name. If a credible set, such as the third one, have most of the SNPs as more than human SNPs, it has less evidence suggesting name causal, then we remove it. And lastly, because even after LD pruning, they could still have overlap when testing these windows. So if we have 2 SUSY credible sets that overlap with each other, we merge it. And in real data analysis, we sometimes need to merge them and sometimes not. And after merging, we take all the credible names and remove the more than human SNPs. And with our simulation, we can test and validate this approach using simulation before applying to real data. The goal of this complicated approach is to reduce the false discovery and get a small subset of SNPs. In GWAS analysis, I define false discovery within this 200 KB region surrounding pruned in GWAS significant mutations. And if there's at least a true causal Neanderthal mutation in simulation within this 200 KB region, it is called a true causal from GWAS. If there isn't any significant name in this region, it's called a false discovery from GWAS. And then we can get a FDP. And in fine mapping, if there's any causal simulated name in the credible names, it's a true discovery. Otherwise, it's a false discovery. Therefore, we have FDP for GWAS and FDP for fine mapping, and we can see how well this approach helps. Again, we have five types of simulations and combined. And fine mapping is shown in pink and GWAS in green. Overall, we can see that fine mapping reduce the FDP quite significantly, with GWAS average FDP about 30%, fine mapping average FDP is about half of that. And when applying to the 96 UK biobank phenotypes, I identified a total of 110 credible regions, which have a set of SNPs for each credible region. And in this figure, I'm just showing you that the power in detecting this credible region is related to both heritability and name heritability. As we can see that there's a correlation between the number of credible name region in the phenotype and the heritability and name heritability within each phenotype. What we really want to see is that after fine mapping, we get a short list of interesting names with potential causal phenotypic effect. And we want to reduce this number of names as much as possible, but because of the strong LD, it's not always possible. So here I compute the number of names in the credible names and also divided by the number of names in this entire region. And that is shown on the x-axis. So in some cases, after fine mapping, we get less than 10% of the names remaining as potentially causal, but in some other cases, the reduction is quite minimal. Overall, that's depending on the phenotype and the credible region and many other things such as LD structure. So that means just purely based on statistical fine mapping, we're still not able to identify a particular mutation, and we have to rely on some other approach. I'm going to show you some interesting example that we identified after combining with annotation and evidence of literature first. And the first example, before I introduce the first example, I want to mention this most prominent functional introgressed region that's identified in the 2014 paper of Amelia, that is for the EPAS1 haplotype. And in this haplotype, the introgressed copy from the Denisovans have elevated haplotype frequency in the Tibetan people. And this introgressed archaic haplotype is associated with the hemoglobin concentration in high altitude, which potentially helped the Tibetan population to adapt to the high altitude. And it also indicated that the Denisovans may have also adapted to the high altitude prior to modern humans. And the reason I want to introduce that is because one of the examples here that we found has somewhat a similar feature as EPAS1. So this STAT2 gene was actually, it has a few mutations in our credible names, and it's actually also one of the first gene that's shown to be introgressed. And in this 2012 paper, the authors show that there's a copy from archaic that's absent in the African population, segregating at moderate frequency in the Eurasians, but have very high frequency, larger than 50% in the modern Papua New Guineans. And the authors also suggest that because this is an adaptive introgression signal in a specific population, it's possibly an adaptive introgressed locus. However, unlike EPAS1, which is now really well understood in terms of biological function and history, it's still somewhat unclear, like why this STAT2 gene have elevated frequency in the Papuans. And as mentioned, we have a few credible names in this region, and this gene has some function that's potentially affecting antiviral activity, and it's one of the family members that's forming a transcription activators. We're still trying to figure out if there's more evidence about this gene and if there's anything else we could say about it. And another example this list is being looked up by Chris, my co-author, and also by Shiran. And another example they gave me from the credible names is this AKR1C4 gene. There are a few mutations in it, and what's interesting about it is that there are two non-synonymous mutations, and they affect multiple phenotypes in the credible names. We only tested 96, and we only have 110 credible regions, but this pair of mutations tend to show up a lot. They at least six phenotypes, and interestingly, there's an old literature that introduced these two mutations into E. coli, and they found that when introducing one mutation to E. coli, there isn't much change in enzyme activity, but when introducing two, there's a strong 3 to 5-fold decrease in activity. And the decreased copy is the Neanderthal copy. So we're also still looking into this mutation. And there are a few other interesting candidates that we're surveying, and hopefully we will share more about that when our preprint is ready. So with that, I'd like to thank Amelia again for having me here, and thank my advisor, Shriram, and all the co-authors for their time and input, particularly Chris and Ali. And this work couldn't be possible without the fantastic UK Biobank resource and volunteers. And thanks for your attention, and I'll be happy to take questions if you have.
Thank you very much, April. I actually have a question for you. So when you were looking at the different phenotypic categories that were overrepresented or underrepresented, do you have any kind of hypothesis on why those specific regions might have been? I guess I was mostly interested in the underrepresented plot. Do you have any thoughts on why, say, anthropometric traits didn't seem to have a lower amount of NIMS in them compared to some of the others?
So we didn't see any enrichment in any of the tested phenotypes, even after meta-analysis. And we find that in this anthropometry part, we have more signal for depletion, which in part could be a power issue, that these anthropometry phenotypes, they tend to have higher heritability, so there's slightly better power for detecting a depletion that's possible. And we don't know. This is just my interpretation that phenotypes with higher heritability may have better power compared to phenotypes with low heritability.
Okay, thank you.
Thanks for the question.
Yeah, does anyone else have questions?
Yeah, I had a question. Thanks for the talk. That was very interesting. I think your analyses, are great, controlling for LD and the Neanderthal or the introgressed, the archaic. My question is a very simple one, is that in regions that are introgressed, where as best as can be told that the entire region is of archaic origin, but contains, let's say, a couple of SNPs in different genes. I actually don't know how long or how many genes are contained in an average introgressed archaic piece. Then you sort of have an internal control for the LD, because you know that the archaic allele must, or that the SNPs contained in the introgressed region must come from the archaic source. Is that, how many of those are the ones that are on the list that have now been cleaned up? You see what I'm saying? I mean, your correction for LD seems great, and obviously a lot of them went away when you corrected. It would seem that if you had a segment that came in and the whole region was archaic and there were functionally relevant SNPs in those, you certainly would be able to infer that the phenotypic effect was from the archaic origin. Are there many of those cases where the
Are
intro-
you talking about the fine mapping part?
Yeah, I guess I'm talking about the fine mapping part. It would have to be at that scale, correct?
Right. So the input to fine mapping is about 200 kb, which I just used 200 kb because after testing a few different lengths, after testing 100 kb. So basically, this region is generally much larger than the archaic introgress segment, which has an expected length of about 50 kb, if I'm correct. So this is like longer than that. And let me pull out this slide. So in SUSI, it takes a really rather interesting approach that it's like an iterative approach of trying to reduce the number of SNPs in the remaining credible set while trying to maximize the regression. And in this case, since we're using a longer region, and also since that even when this region is only archaic, it's not 100% frequency, it's still archaic mutations are at low frequency. There will be a more than human haplotype. So regression will still work, even though there's strong LD among the archaic mutations and strong LD among the human mutations, but it's like two semi-unlinked group of SNPs. It's just human mutations tend to have low LD, but archaic mutations tend to have high LD. So it's very hard to have a small subset of SNPs that is only archaic because they're so strong in LD, say 0.99 or higher than that, then there's no way of further decrease the size of this credible set.
Right. Okay. That answers the question. Obviously, in super high LD blocks, you can't tell which SNP is causal. But obviously, if you had an archaic SNP that was somehow in LD with a modern human SNP, you can correct for that and then
Right.
reassign that, which you've already nicely done. Okay. So that answers the question. Yeah. So a lot of the LD within entirely archaic regions is extremely high between SNPs. Yeah.
Yes.
Great. Thanks. Nice talk. Thank you.
Thanks, David.
Any last questions? We probably have time for one more if there is one.
I had a question. Thanks for the really great talk, April. My question was, how did you choose the phenotypes that you analyzed? Because I noticed that that there weren't any. It didn't seem like there were any traits related to the brain, which previous papers have reported on young cell introgression and brain-related traits. So I was just wondering how you chose the 96 traits.
Okay, that's a great question. So we have a number of phenotypes that have been studied in Sharon's group and also a few other groups that people define as interesting. And I took all the phenotypes from there and then removed the redundant ones and highly correlated ones, and which result in this subset of phenotypes. There are and also remove the phenotypes that are not phenotyped in a lot of individuals with a threshold. So basically, like if a phenotype is only measured in 5% of the UK Biobank individuals or 10%, we're not going to have a lot of power. And those are the criteria. But the initial part is the list of phenotypes we have access to, and then and then I hope that answers your questions.
All right, yeah, thank you.
Thanks.
All right. Well, thanks everyone for joining us today. And thanks especially to April. And we'll see you next time. Take care, everyone.
Thanks. Bye.